It is with great pleasure and a real honor to introduce to you Dr. Sam Broder. Dr. Broder has had a very distinguished career. He has been appointed by President Reagan as director of the National Cancer Institute, a position that he held for 6 years. His laboratory interests included antiretroviral therapy and the relationship between immunodeficiency disorders and cancer. In the mid-1980s, his team developed the foundational antiretroviral drugs now widely used in therapy of AIDS and related disorders in adults and children, including AZT and Videx. How do you pronounce Videx? His lab also oversaw the development of various anti-cancer agents such as Taxol. As director of the NCI, he helped launch a number of large-scale randomized clinical trials related to prevention, diagnosis, and treatment of cancer, especially breast cancer and prostate cancer. In 1998, he joined Celera Genomics at its founding as the chief medical officer. His contributions have been essential for the success of the Celera team in sequencing and assembling the human genome. He led the Celera team towards applying the new genomic discoveries to the evolving technologies towards personalized and targeted medicine. Dr. Broder is the author or co-author of 340 publications, scientific publications. He is a member of the National Institute of Medicine and has received numerous scientific awards related to his research in cancer and AIDS. For many of us who had the pleasure to work with Dr. Broder at Celera, he was clearly our mentor. Who can forget our passionate debates on the multi-hole problem, the difficulties of the use of conditional probabilities, especially related to fallacies in clinical trials. We can forget the fundamental cultural changes that needed to take place in the FDA practices once the human genome was made available, or our passionate discussions about aspects of game theory and pharmacoeconomic behavior. Our Professor Broder, with a command of mathematical critical discourse unusually strong for a doctor, taught us and inspired us to invent new computational methods toward genomic discoveries that would lead eventually to a practical impact on clinical applications. It's a great pleasure.
Thank you, Soren. I really appreciate it, in particular for your formidable achievements at Celera and before and here at Brown. We are about to enter the 25th anniversary of the first patient who received AZT and related antiretroviral therapy on my service at the clinical center. And that occurred in July 3rd of the year 1985. So this coming year, 2010, will be the 25th anniversary. And I want to take this opportunity, this privilege, to talk to you about some of the issues. Some of my talk will be historical in that context, looking back in the era and to see what lessons we can learn. And some of my talk will be based on hopefully planting ideas that could be implemented for the future. In fact, hopefully ideas in the realm of computational biology that might speed progress even further. So the title of my talk today is The Development of Antiretroviral Therapy and Molecular Diagnostics and Their Impact on the Global HIV-1 AIDS Pandemic. And I subtitled it Lessons in Treating the Untreatable. And so hopefully we can talk about... This was a slide that I prepared in the year 2001 from a lead article in Morbidity, Mortality Reports, and also the New England Journal, where AIDS was described as entering the third decade. We're just about to enter the fourth decade. And so in that sense, we have an enormously longer journey to traverse, but in other senses, we've come a long way. I like to show this slide because several issues related to it would have been impossible to conceive or would have led to out-and-out derision had it been proposed in 1985. And so this is an article from a relatively recent editorial, and I've amplified some of the key sentences. But the title of it says it all. That is, there, at least in the initial treatment of AIDS, HIV-1, there is an embarrassment of riches. In fact, incredible though it may appear, there actually are prioritization issues in designing future clinical trials for the primary initial therapy of HIV-1, not because there is a dearth of effective interventions, but because there is an extreme abundance. That in no way implies that we have perfect therapy or that the disease cannot be formidable. But there is a range of very safe and effective therapeutics that have evolved, and there is no reason to believe that that evolution will not continue. And so taking a sentence from that same editorial, with more than 20 drugs to choose from, actually there's probably more at this point, there is an embarrassment of riches. Possible combinations are almost endless, as are the possibilities of side effects, either beneficial or damaging drug interactions and the development of viral resistance. So that, in effect, is also a warning that while we have made extreme progress at one level, and while the progress, I personally believe, has exceeded any of the more common so-called realistic projections 25 years ago, there also are ominous signs that we should not take this level of progress for granted. What I'm going to show here in the next few slides, and the actual specifics are not critical, but I'm showing you products that have been approved by the Food and Drug Administration in the United States. And there are approximately 30 products that have been approved either alone or in combinatorial form. And many of these in fixed dose combinations have been adapted to the developing world, to resource poor countries, but the totality are available in resource-rich countries. And basically, there are nucleoside reverse transcriptase inhibitors. There are non-nucleoside reverse transcriptase inhibitors. There are protease inhibitors. There's even a fusion inhibitor. There are entry inhibitors. There's an HIV integrase strand transfer inhibitor, and probably others on the way. And there are multi-class combinatorial treatments. So we have an enormous degree of progress. Are there any physicians in here? There are 2. Do you see 3? Do you see AIDS patients? May I ask, when was the last time you saw in your own first-hand visual inspection a patient like this? Have you ever seen a patient like this?
Not that bad. Probably less.
So within the last year, have you seen any patients like this?
No.
Okay. I recently participated as the introductory reviewer, author for a soon-to-be-published review collection of an anthology of papers, and to discover all aspects, I'm writing the overview. And as I'm writing it, I'm looking at the author list of what is going to appear in the same edition. And I noted, there was no description of Kaposi sarcoma, which is what this is, obviously. This is fulminant Kaposi sarcoma. And I said to myself, boy, is this progress. They don't even consider this important enough to include a chapter on it. At any rate, I showed this slide. This was a patient from my service at the clinical center. I showed this slide as a mnemonic device to remind you that at the time, AIDS in 1985, 1 year after Gallo had published his formal proof that HIV, a novel retrovirus, was the cause, not an incidental passenger, not some agent in contention, but the cause of AIDS. Gallo and his coworkers, this was the kind of patient we were seeing. We were not seeing patients with minor illnesses. We were seeing patients who had a lethal and terrifying disorder. And in most cases on our service, it was fulminant. They were fulminant opportunistic infections, usually combined with some, on our service at any rate, a very serious and often lethal malignancy in the form of either disseminated Kaposi sarcoma or aggressive lymphomas, often of the central nervous system. For those of you who are clinically inclined, you can see the Kaposi sarcoma, but if you're very careful and look at the flanks of the patient, you can see severe edema that is not fat. The patient had enormous fluid accumulation and so on in consequence of the disease, and in fact went on to die of this as the primary cause of death. The Kaposi sarcoma, as we now know, is caused by an unusual herpes-like virus, HHV8, and is probably linked to the severe immunodeficiency. But what I'm pointing out is that this disease, one of the stigmata, original defining stigmata, this component, has not literally disappeared from the United States, but has become very, very uncommon. And that is also a sign of progress, which I'm going to show you. But I'm using this slide to remind you that we were not treating minor cases or people that were well carrying the virus. We were treating people with fulminant disease. And here are the issues that we faced at the time. I boil them down. There are many subsets of this, but in this particular context, I'm going to talk about essentially 5 obstacles that existed at the time to an effective response to HIV-1 AIDS. One, active or replicating retroviruses did not exist, were felt not to exist in human beings. These are all obstacles or belief systems that were in opposition to progress, in my view. Then there was another philosophy that, well, even if we concede that active retroviruses exist, if they did, they were not the causation of human diseases. In other words, okay, you can find some retroviruses floating around, but they don't mean anything. Then the other 3, in the alternative, for people that didn't want to deal with 1 or 2, even if somehow active retroviruses did exist and cause disease, the agents played a relatively minor anecdotal role in the public health and were limited to unusual subacute T-cell leukemias or unusual neurologic syndromes, as in the case of HTLV-1 or 4. Even if somehow those first 3 conditions somehow did not apply, retroviruses by their very nature were inherently untreatable, and that is based on primarily on their capacity to integrate into the host genome and or rapidly mutate due to the error-prone reverse transcriptase. And then early after the discovery of HIV-1, there was an exceptional diversity recognized of this new virus. And that was a cause of great concern in its own right. So even relatively early, people were noting that there was an unusual diversity to this virus. It had a method of diversifying, mutating, that was greater than the average viral pathogen. And most retroviruses at the time, and this one I think was pretty fundamental, most retroviruses at the time were unfamiliar with drug development, and they believed that a vaccine was the higher priority in any event. So if you believe in a theory of alternative universes, one could argue, supposing you have a universe where the retrovirologists who believed that a vaccine was the only logical route, supposing in some way they had prevailed, supposing that they were on study sections that determined research and funding priorities, you can draw an alternative universe of what might or might not have happened. And I think asking that question and positing it that way. I also want to point out that retrospectively everything is easy, and it is very simple to argue that a particular hypothetical fear would not have materialized. It is very easy to say, of course, people would have been open-minded and study sections would have allowed this or that. But there are issues with that. I never subscribed to this view, not because I believed that retroviruses were inherently impossible to treat. This is my timer. I guess I'm done with my presentation. So at any rate, I never subscribed to that view, not because I felt that vaccines were impossible, but I felt that it was basically unwise to predicate an entire program only on one pathway. I also felt that a vaccine approach for primary disease, as I mentioned to an earlier group, had a certain element to it that was quite unsatisfying. In other words, at that point in time, we knew that tens of millions of people, some estimates 40 million, had already been infected with a lethal retrovirus. And if, in fact, one said, we are going to develop a vaccine, but it's only for primary protection, well, you're leaving 40 million people to their fate. So I never quite got the dynamic of that, to be candid. And I never understood it even to this day when people say something which is a truism, that vaccine is the definitive answer, which of course it is if you condition the question the right way. But on the other hand, one, you don't have a vaccine really that's operational, so it's a hypothetical. And two, you still have to address what do you do with people already infected, unless you're going to invoke that you're developing a therapeutic vaccine, which is even a higher level. So I think these 5 points will allow you to take an orientation as to where we were when we started. Our philosophy was basically that AIDS was a lethal and terrifying disorder. We needed to do something. We could not wait for a perfect answer. Also, we were in the Cancer Institute, which I'm going to bring out a little bit, and also in the intramural program, which for those of you who may not quite realize what that means is we had a laboratory and a clinic under our own control. That is, I had a lab, and I also had a clinic a few feet away. And so we actually could move from the lab to the clinic, and were, at that era, encouraged to do so. I think that one of the issues we need to think about is whether we are creating a division of labor in clinical investigation in terms of having basic researchers and having the clinical investigators who really have a totally diversified and differentiated kind of activity profile without having at least a small number of people who do literally do both for a living and are charged with doing both. I personally think that if there had been a division of labor like that and it was highly enforced, there would have not been drug development, at least in my laboratory, and probably there would have been significant delays. Because committees are not necessarily the optimal way to generate pathways that have not previously been undertaken. And retroviral therapy at that time was considered impossible. Okay, I want to show this slide, which I'm going to show again. This slide is an intuitive phylogenetic tree. The distances that you see, what you see is what you get. If things look like they're diversified in this slide, they are. So what you have is an example of what some of the issues were. This is influenza in 1996, but it's typical for any influenza. This is influenza globally at any one time. So it's diverse, but not really very much. It's true that influenza will change with time, but at any one snapshot in time, this is the diversity you're dealing with. Look at the HIV of an individual. That is the diversity within a typical individual at a particular snapshot in time. This is an epidemic of a subtype, a beta subtype in HIV in Amsterdam. And look at the diversity that you have there. And then look at the Democratic Republic of Congo and other parts of West Central Africa. And what you have is what you intuitively, you are seeing what the diversity profiles are here. What you can say is that a particular HIV in a particular individual has as much diversity in an individual as influenza does globally at any one time. This level of diversity was actually suspected or known relatively early. And yet it was another complication that people felt as to the futility of doing too much with HIV-1. That something that is this diverse is not going to be amenable to interventions. That is a picture of me approximately that time, before the weight of the worlds and responsibilities, before I realized the depth and severity of what failure could have meant. And I was pretty optimistic and cocky at that time. This is the first paper from my laboratory on AZT, 3'-azido, 3'-deoxythymidine. And I did a Socratic method with one of the students that was here earlier as to how this works. Actually, these are, in this class of other drugs that we studied, are glorified Sanger sequencing reagents, in effect, in my view. That is how they work. The reverse transcriptase of the virus likes them better, has a higher affinity, will incorporate them, will tolerate them better than the DNA polymerase alpha, particularly alpha. We can talk about DNA polymerase gamma to some extent later, by significant differences. And if the host cell has the relevant phosphorylating enzymes, and this is crucial because HIV-1 does not bring in its own kinases or activating enzymes, unlike herpes. So the host must provide all the anabolic phosphorylation, all the activating enzymes. And if the host cell does, AZT or other drugs in this class, which we worked on, will become anabolic phosphorylated, activated, and in effect, we believe they serve as Sanger sequencing reactions to the virus. The virus incorporates them in growing strands as it's the reverse transcribed DNA is being elongated or the second strand is being elongated. Once the false strand is introduced, that chain is dead. It will either be destroyed or whatever. And the virus does not have effective repair. So that's AZT. This is its structure. For those of you that are medicinal chemists, there's an interesting azido formulation here. This is an old drug, ironically. It was first synthesized in the mid-60s by Horowitz and his coworkers as an anti-cancer agent with an NCI grant as a potential candidate anti-cancer agent, and it wasn't toxic enough against cancer. And was essentially languished in obscurity for many years. So this is another paper by Mitsu and myself that really defined the structure-activity relationships, and we published that very soon after. And this is, we basically learned what turned out to be a relatively simple structure-activity relationship. I call it simple now. I don't mean any false modesty or even sincere modesty. I think it's simple only in retrospect, because what we now know is that you need a nucleoside base. You need to have these two in either this in a deoxy or in this at a block position, or some equivalent. We'll talk about, if we have time, a few minor variations on this. But those are the basic rules. They're not enough. They don't guarantee that you'll get activity. But at least in that era, those were the drugs that were candidates. And a number of drugs came from that idea. This is the first clinical trial for my group, and it was published in March of 1986. So to give you a scope of how fast things were moving in that era, the paper on AZT, this paper was submitted in June of 1985. This is the laboratory observation. This paper is the first clinical results where we thought we were seeing clinical activity, and it was published in March of 1986. The drug was approved in March of 1987. So I don't believe you can move much faster than that. And we'll talk about some of the issues. We were talking in an earlier session about what are the impediments to drug development. This, ironically, was the first patient of AZT who presaged virtually everything. In one of those things where we thought, my God, we are now, having lectured the world on ascertainment bias and selection bias and publication bias, we're now going to face that ourselves, because the very first patient had a six-fold increase in the CD4 count by week 2, was anergic and converted to PPD. There were other stigmata of improvements and other indicia that the patient was getting better. And we, of course, were trapped in the paradigm that it's an open-arm study. You have to be cautious what you say. Maybe some of these effects are spurious or some kind of regression to the mean or something. But it turned out this first patient had everything that was subsequently, ironically, that was subsequently confirmed by randomized trials. And this was the randomized trial. I did not do this trial. This was done at a multi-center trial. But it was very instructive because it was an extremely difficult opportunity. It was a placebo-controlled trial, essentially using death as an endpoint. There's no way of sugarcoating it any more than that. And we received enormous criticism for this particular study and its implementation from both sides. There were many patients who believed that it was improper to do a placebo-controlled trial for a disease like this, where people at a randomized level were going to get a placebo. And there were others who believed that this trial should have been permitted to continue. It was effectively prematurely terminated when a data safety monitoring board showed an enormous differential in the death rate in an arm of the study. And when it was broken, it was found that was AZT was significantly protecting survival, and the placebo showed a very high death rate within a relatively short time. So the study was stopped prematurely, on a safety board determination. But there were people who believed that the study should have been allowed to continue longer, which are mutually contradictory points of view. I think, in retrospect, the study was necessary, and it was handled precisely as one should have handled it. I think it would have been impossible, from everything that I know, to reach a consensus ever on the activity of AZT, and then by implication any antiretroviral agent, without having a randomized placebo-controlled trial. I think it would have been very difficult, and I think that would have induced interminable arguments, not only about whether any antiretroviral drug worked, and therefore effectively deny patients in the future safe and effective treatments. But I also think that on balance, it would have effectively played into and revived, extended, and amplified a debate by AIDS denialists who believed that HIV-1 was not the cause of AIDS to begin with. So I think there are many different complex reasons why this trial was exceedingly important, why it was conducted, because for many reasons, not the least of which is that AZT, because it's a chain terminating nucleoside, could well have, in theory, hurt patients more than it helped. One didn't think that that was going to be the case, but in doing a randomized trial, sometimes one finds that a particular agent actually is toxic, and that's one of the functions of a randomized trial. So there was enormous controversy over this point, but I believe it was essential, I believe it was the right thing to do, and I believe it spared many, many people who came later from the uncertainties, the complexities, and the toxicity of a lack of knowledge, because a lack of knowledge is a form of toxicity. And so the study was stopped. Both arms immediately began receiving AZT, and I think that was the right thing to do. I think the issue with this is also laying the groundwork for some points I'm going to make in a minute about the importance of surrogate markers and molecular markers. The development of AZT occurred before PCR was invented, as shocking as that may be. There were no tools other than viral culture to monitor, and there were no surrogate markers, which is another reason. Unless you wanted to argue CD4 count could have been a surrogate marker, but most people were extremely unwilling to accept that at the time as a surrogate marker, and perhaps even to this day. So these are some other findings that we had. Again, we lived in a parallel universe at the time where there was enormous, particularly in the space of 1985 to 1987, where there was enormous pessimism based on those initial postulates that I gave you, why things would not work, including that there might be excessive toxicity to the drugs and so on. While in our own internal service, we were seeing significant benefits. And the team that I was leading at the time was primarily made up of Hiroaki Mitsu, who did the lab work, and Robert Yarchwan, who led some of the clinical studies. But the point that we were getting in that era was that we were seeing improvements in patients on a wide scale, including reversals of dementia. So there was an additional complexity of this issue, because not only were we essentially suggesting to our colleagues that HIV-1 could be treated, which they found in itself inherently impossible, or at least very difficult, and many did not accept our preliminary subjective findings because they rightfully thought there would be a bias attached to it, or we had an unconscious ascertainment bias or something. But they also, on top of that, had an amplified reason for skepticism, because we were asserting that you actually could see reversals of dementia or other neurological features. Bear in mind, this was an era where antiviral therapy against a virus of any kind, never mind a retrovirus, was still very novel. And then if you're invoking on top of that we have a chronic virus of the brain, and we're somehow able, on essentially what amounts to one of the first times up at bat, reverse the dementia, or at least temporarily hold it at bay, that added extra complexity and put us into a parallel universe where we were optimistic and almost everybody else around us was extremely bummed out. So this was a study that we did relatively early, and we found that there were, okay, well, maybe we're good. We're good educators, and we can take patients that are demented and train them to look like they're not demented, or people that are in a coma and wake them up. I mean, maybe we were good, but we felt that these were quite objective glucose metabolic utilization studies using positron emission tomography. And you could see in a before and after kind of thing that you don't need to be a radionuclide expert to see the difference. And the patients were having significant improvements in their brain metabolic function, which we did not think was an artifact. We also saw changes in young people with evidence of cerebral atrophy. This was a patient, as you can see by imaging, has large ventricles and so on. And then by the time they've been on therapy for about 2 years, they have significant improvement, clinical improvement, but significant improvement on imaging. Now, one of the points I want to make is that many years after the antiretroviral authorities, I became the director of the National Cancer Institute. And in that era, we had the first round of health care reform. And one of my duties was actually to escort high-ranking people. So at this time, this was the highest-ranking government official there was. It's a little joke, but not too far off. And basically, we had health care reform. And so I was happy to take everybody around. And then, as now, I was struck by the following. There is very little discussion. I am not saying there is no discussion, but there is very little discussion of research, basic and clinical, as an element to healthcare reform. It's an odd omission, in my view. That is, everything is sort of predicated on comparative effectiveness research, and I'm not dismissing the importance of these concepts, but the issue is basically almost framed as though we have a total universe of knowledge. It's already in a box, and all we have to do is sort out the priorities of what works best. And the notion that we have to generate new knowledge on a continuing basis, and that is part of healthcare reform, a legitimate element of healthcare reform. How do you generate new knowledge from basic research? And how do you develop the basic research seems not to be high on the agenda. I don't know if you get that impression, but I certainly got the impression the first time, and I'm living a deja vu of that again, that healthcare is not viewed as, if there is research, it's in a very specialized area. There's also a subtext, a thinly disguised subtext, that research generates extra costs. It creates new technologies, and they cost a lot, which may or may not be true, but my only point is the notion that basic research can solve issues and render whole new paradigms, this seems not to be a high priority. And I think that one of the reasons that I like to emphasize the HIV story is it's a living refutation of that. So, this shows the changing kinetics of the pandemic in the US. And I'm going to come back to variants of this slide. But this gives you different kinds of milestones. So monotherapy with AZT was essentially introduced at a significant scale in the late 80s. There was combination therapy was introduced. There was a reference lab HIV viral load assays, FDA approval of HIV viral load kits, and for both prognosis and monitoring. And each of them played a role. But there was also wide-scale use. For example, once the viral load kit was available, it was already recognized it was more than prognostic, and doctors had already adapted. I'm going to come back to this notion. So what it's done, what we have now is also molecular tools for determining viral resistance, in particular as it applies to the reverse transcriptase and the protease. So in effect, many doctors have now become molecular biologists. And this gives you can actually genotype the virus. You can come in and define a mutation, and you can say, oh, you need to be switched to this or that drug or so on. In other words, it's become a perfect paradigm for personalized medicine in an extreme degree. You identify the specific molecular lesions that are going on in a particular patient's virus, and then you choose from a menu of therapeutic options wide enough to encompass what you can do at a clinical level. I show you this slide. I've shown you this slide before, and as I mentioned earlier, this is one of those slides that I never tire of looking at or showing. What am I showing you here? I'm showing you the age-adjusted rate of death due to HIV in the United States. And what you can see is that there was, during the 80s, a dramatic, and even before that, but we're just going to show, a dramatic increase in the death rate at each year, and then a sudden and precipitous drop. Not accompanied by a drop in the incidence, not accompanied by a paradigm shift of a new vaccine, not accompanied by a sudden cessation of the pandemic. I mean, you can see cessations of certain diseases when a very dramatic new intervention occurs. We didn't suddenly become super effective, and that wouldn't have mattered anyway, because it's a chronic virus. You can't turn it off suddenly even if you were suddenly to stop new infections, you still have a pipeline of old infections that are going to work their way through. So this was a precipitous drop in the death rate. And what happened in this year building up to this? What happened is none other than the introduction of highly effective antiretroviral therapy in combinatorial form. Now, ACT was endowed, for those who believed in it, with more that could be done than could be done by any one agent. When AZT was approved, there was a tremendous expectation that it was the answer. And it was very difficult to explain to people that, wait a minute, it's just a start. You can't rely on one drug alone. But before you have combinations of several drugs, you have to have one. And you can't have 5 drug combinations until you've had your first one. And so the whole idea of introducing combinatorial therapy, highly active antiretroviral therapy, took time. And one of the things I was talking to the class that I helped teach a little while ago was, what if you were a planner, an economic planner, a medical economic planner, and you were trying to prioritize things? Would you have logically said at the cusp there, let's say in 1994, that it's time to throw in the towel, that the rate of death of HIV is going up, it's unabated, everybody is doing antiretroviral research, it's great, take a victory, non-victory lap, but we're not funding this anymore? Because you're not going anywhere. So how do we plan for things like that? How do you plan for sudden improvements? And that becomes a real challenge. But I show this because it is a quandary that one does sometimes have to prepare for significant success in opposition to what the data seem to be showing. By the way, what I just showed you a minute ago was the total population. Here is focusing on people just in the 25 to 44-year age group, which was a particularly hard-hit group with HIV. And you can see that briefly, HIV became the #1 leading cause of death in this age group compared to everything else that the National Center for Health Statistics keeps statistics on. The other thing is that from time to time when I show these data, somebody comes up to me and says, how do you know that some systematic error didn't come in? Well, these are based, by the way, on the National Center for Health Statistics on death rate certificates, on the actual death certificate. And there were no significant variations year by year for other causes. So at any rate, this is another feature that we need. So why was antiretroviral therapy? Is there a lesson you can learn from it? And this is one of the points I'd like to make. Why was it able? Why were we able to take a virus that was thought to be inherently untreatable in an era where barely antiviral therapy of any type barely existed, and yet ironically it was possible to develop a series of drugs, a cornucopia of drugs that are active, at least in primary disease, and in many cases to be able to say that you're converting HIV, which is a lethal and terrifying disorder, into a chronic, manageable condition. How did that happen? It seems ironic. And I think there were many factors that we could get into, but I want to focus on the one factor that I think has helped enormously, and that is the development and acceptance of reliable surrogate markers of disease. We do not have that for a lot of diseases. When HIV viral load, however, the RNA viral load, became recognized as not only a prognostic factor, but actually as a reliable surrogate marker, that was a revolutionary change. And it made it possible to select or deselect new therapies or drug combinations very, very fast, and to bring in new drugs, to keep them, and to have a high expectation that if something suppressed viral load, it was going to have an effect at a clinical level. I'm using the term surrogate marker in the following way. My definition of a surrogate marker is that you can rely on it as well as clinical endpoints, just as well, to reject the null hypothesis in a clinical trial. And that when you use the surrogate marker, you get the same answer as if you use clinical endpoints. And that is a rigorous definition, but I think actually applies essentially to the HIV RNA story. And with the use of that, it became possible to do a range of things, including, for the purpose of discussion, really monitor a drug for virologic failure, but also have a commitment to monitor new drug development. And so a sponsor of a new drug, a private drug company, could come in and have a reasonably clear pathway to whether it could get a drug approved or even rejected, because the scenario that a pharmaceutical company doesn't want to be in is having to commit to a study and then not be sure when it's time to get out or get in. And so it really becomes very difficult if you are committed to long-range clinical studies with no intervening surrogate marker. And in the case of HIV-1, I think having a surrogate marker changed everything. And basically, I think it changed a great deal in the drug development process. So at this point, I want to switch gears a little bit and talk about really what AIDS was all along de facto, but really has become. This is a picture from a UN publication. And in subtext form, I think you can see that what they're trying to tell you is on the right-hand side, there is a grandmother. And on the left-hand side, there is her grandchild. Neither looks happy. And the faces express the faces of significant trouble and bordering on despair. But this is the face of HIV as it now exists, and the epicenter of the disease is in sub-Saharan Africa. And the disease, the reason I think this is an interesting slide to use is, in effect, the face of AIDS now is the generation, is the eclipsing of productive members of society in a given generation in many countries, where relatively productive young adults become sick or die or incapacitated. They leave de facto orphans behind who have to be cared by elderly survivors in countries that have almost no resources anyway. And so there's a cycle. So the economics of the country get damaged because their productivity of their workers is gone, and there's a cycle. And that leads to multiple issues that I want to raise that I don't see often raised, but are part of the equation. So this just gives you a summary. As of now, there are probably 33 million people living with AIDS. I want to just look at this slide for a minute. If you look at what the adult prevalence on average, they're listing it at 5% in sub-Saharan Africa. We're within range of that in the District of Columbia. So this is not only a developing country. I believe that I've seen statistics that up to 3% of certain population groups are infected. So I think it's not only a problem of resource-poor countries, but I think sub-Saharan Africa is the epicenter and has certain very serious implications. In some countries, the prevalence rate is higher than 5% by far. This is just a general average. And I wanted to introduce this notion that AIDS is part of the story in which there's a correlation between diseases and civil instability or unconstitutional changes in countries. That is, that they will lead to armed conflict or destabilization or coups d'etats and so on. And it's hard to separate cause and effect because sometimes the particular infectious disease is both cause and effect. So here's a quotation from a UN report that AIDS and global insecurity coexist in a vicious cycle. There are civil and international conflict help spread HIV as populations are destabilized and as armies move across new territories. And basically at the end, the end sentence is really the most key one. HIV/AIDS is both cause and effect, initiator and beneficiary of instability and conflict. So I don't see this discussed very often, but the notion that AIDS is part of a broader issue of infectious diseases that actually help destabilize countries. And so this is beyond the normal boundaries of public health. Critically important though public health issues are, that basically the level of instability that can occur by something like an AIDS pandemic can lead to other issues. And this was a slide that I created in a publication that I helped put together right after the human genome was done, under the theory that the human genome could open up a new opportunity for applying high technology in solving problems like malaria and other infectious disorders that were part of the issues that we're talking about. This shows a regression, if you will, for the percentage of countries that are engaged in armed conflict versus the number of children who die of childhood diseases. And I think there's a pretty good regression there. So whether it's cause and effect is always complicated, but I believe it is both. The same thing is both cause and effect. And this is a slide that I put together as part of the same paper. I don't want to overstate this case. I don't want to make more of it than I think there is, but there is a lot. And what these little symbols represent are basically where armed conflict exists, and then you can see where things like tuberculosis or childhood mortality or even HIV prevalence are significant. And there is, at least for food for thought, a concordance that, again, gets you into the issue, which comes first. But I think in this case, it's both. It serves both. So I want to talk about the future challenges to the current paradigm of antiretroviral therapy, because we've talked about some of the issues as to how we got here. And ironically, to me, one of the largest issues that I never thought I would personally live to see the day is complacency that the problem is solved. And part of it is because, in my opinion, in the United States at any rate, HIV-1 mostly is no longer considered a lethal and terrifying disease. Do those of you who are physicians agree with my assessment? Basically, there are things you can do. When we had patients, when we were first starting off, patients would come in, they would die within weeks. And there was nothing you could do. So at any rate, I think there's a feeling that, okay, it's a serious problem, but we can take care of it. And that may be true, but it may not always be true. And I'm going to talk about that basically being vigilant and being hopeful are not necessarily contradictory. So here's some challenges. Integration of the provirus and an unpredictable viral latency. The viral rebound will follow discontinuation of treatment in any of the current paradigms. And therefore, you're basically talking about treating a patient for a lifetime. We're going to talk about some ways of getting around this, including maybe some novel applications of small RNA molecules. And we can come back to that. Drug resistance, including increasing genetic diversity of HIV-1 and the ongoing risk of cross-species infections with new pathogenic viruses. Well, resistance to many drugs has already emerged. Fortunately, in an odd way, we've been able to keep up with the resistance, and it hasn't become an intractable problem yet. It has in some communities, perhaps, but it seems that one new drug is always introduced with a higher genetic barrier. So in a way, to resistance, we've been saved, in a sense, by the developmental programs. But the current paradigm relies heavily on targets that are encoded by the viral Pol gene, that is basically reverse transcriptase and the protease, and to some extent the integrase. So we need drugs that are beyond these canonical targets. In addition to that, non-B HIV subtypes, or clades, are becoming common. As many of you know, the B subtype is the predominant type that's found in the United States and Western Europe. But in Africa and other parts of the world, it's non-B subtypes. And these non-B subtypes may not behave the same way. They're becoming introduced into the US. One of the notions we have to keep in mind is anybody in the world can be anywhere else in the world, probably, with at least any other population center within 24 hours. So that's a significant issue. The other thing to keep in mind is that it may be these other strains, in addition to classical drug resistance, these other strains may not be detectable so easily by classical viral load assays. And so one has to be able to design assays that have tolerable elements for mismatches and that accurately report all known group M, O, and N viruses. Now, some of the commercial tests that are available only report B. So if we get these other strains, I heard a person say, well, I don't see that as a problem, without realizing the futility of what they were saying. Because the person effectively was saying, I don't see that. Well, excuse me, if you have a test that doesn't detect what you say you're not seeing, how do you know you're... It's a kind of a circular thing. At any rate, I think that's a problem. It has to be evaluated. But equally, there's also a reservoir of retroviruses in non-human primates, and they could create new opportunities for cross-species jumping. As many of you know, the three most common groups of HIV-1, M, N, and O, occurred probably through independent jumps from the chimp equivalent, simian-like retroviruses, mainly probably with group M being the most diversified, occurring as a founder infection approximately 1930. So we're not talking ancient history. We're talking relatively recent. The other thing to keep in mind is that in addition to these three independent transspeciations, we appear not to be done. There was a recent case of a Cameroonian woman living in Paris who had had a transspeciation infection from a simian retrovirus that exists in gorillas. And they designated this new retrovirus as the founding member of Group P. The problem with this is that the regular tests that are often used to detect it fail to pick this up. Not all commercial tests pick this up. And so one has to be careful and make sure that the commercial test is really capable of picking up these new strains. Otherwise, you're going to miss them and the problem won't exist for you until it's too late. And this woman was said to be asymptomatic by the authors who made the publication. I went to a recent meeting and I pointed out to them that they reported that the patient had a CD4 count of 300. Maybe she was technically asymptomatic, but that was a dramatically low CD4 count, and I therefore would not conclude that the virus was inert. They seem to have agreed with me because the word got back that she was treated. So even though they thought she was asymptomatic, they acted. There are cardiac and metabolic combinations. I think this is a very significant issue. Even though a very prolonged survival is now possible, several of the agents actually cause insulin resistance syndromes. They may cause changes in redistribution of adipose tissue. They can cause lipodystrophy, and they can also cause certain cardiac effects. So I think what we may be moving into possibly in the future is that we will try to look at the genetic endowment of the host and other risk factors of the host, not the virus, to make assessments as to what drugs one should give. And it may be that in addition, the average infectious disease doctor is going to have to be very alert to modifiable cardiac risk factors. And the irony is that many people who went into infectious disease weren't trained to do that, but they're now going to have to be trained. They're going to have to be highly focused on some of these issues because as patients live and as these cardiometabolic side effects become more important, that will become an issue. So we talked about this earlier. We ourselves at Celera have made a discovery on a novel kinesin protein that seems to be a risk factor and seems to control statin responses beyond the classical lipid profiles that one uses. And that, I think, gives an issue and gives an example of the kind of things we may want to talk about. Pre-exposure prophylaxis. I think there is a very interesting opportunity for pre-exposure prophylaxis. And basically, some of the second or third generation nucleoside or nucleotide inhibitors can be combined essentially into once-a-day formulations. And the odds are strong. I can't say they're guaranteed because only a clinical trial will determine that. But I believe personally the odds are strong that this approach will actually serve to prevent the transmission of HIV-1. There's a very significant mouse model where the mice have a humanized immune system and non-traumatic vaginal installation of HIV-1 in these mice, to recapitulate one key form of human transmission, was completely blocked by, in this case, a parenteral administration of these two drugs, tenofovir and emtricitabine. So these are two drugs that are both approved, and they can be put together into a combination. And they might particularly empower women in the developing world where there's coercive sex or where the woman cannot make her or induce her partner to use a condom. And therefore, there may be opportunities for chemoprophylaxis along those lines. And I think this is a very important opportunity. We, in effect, have had a kind of pre-exposure prophylaxis in the form of treating infants from mothers who choose to breastfeed their infants, in which protection of the infant is possible by treating the infant for 14 weeks or so with combinations that include AZT. And that's a form of pre-exposure prophylaxis in my definition. So there's actually been a proof of concept of this novel idea. There's also a modified version of this general idea called screen and treat, in which it may be possible that certain countries would implement programs in which either serodiscordant couples or general population would be screened for new infections of HIV. And then upon the conversion, individuals would be asked to take therapy, not necessarily to protect themselves, though that probably would happen, but also just to drop the viral titer down so they become less infectious. And there are mathematical models where that would have a dramatic effect on AIDS. I think we also we need to talk about new paradigms And I have a few to suggest here. There are many reasons to believe that proteasomal inhibitors could be theoretically adapted. You may be aware that several viruses, including HIV-1, by convergent evolution, have adopted the notion of targeting certain host cell restrictive factors for degradation by the proteasome. HPV seems to have learned that lesson, right? by targeting P53 and RB through its own proteins, targeting them for proteasomal degradation. And what happens in the case of HIV is the viral accessory proteins VIF, VPU, and VPR are all thought to connect to modular ubiquitin ligases that bring about the polyubiquitination and proteasomal degradation of the cellular host restricting factors. In other words, there's been kind of evolutionary biological warfare between humans or human precursors and retroviruses. And the virus does get smart, and then the human develops an endogenous innate cellular restrictive system to counterbalance it. And then the virus gets even smarter, and it goes back and forth. In the case of HIV-1, it involved some so-called accessory proteins that are deliberately designed to target certain host restriction factors. So if you have a proteasomal inhibitor, it may be possible that you could dose it, maybe at small doses. In the case of VIF, which is one of the key viral accessory programs, this could protect a very important target called ApoBec3G, which has certain functions that effectively cause an error catastrophe in the virus. It's kind of a cytidine deaminase, and it is thought, at least as one mechanism, to hypermutate the virus as it replicates and essentially stop the virus through hypermutation. And the virus says, I don't like that. I'm going to target this for degradation, so you do not mutate me to death. and if you have a proteasomal inhibitor, you may be able to overcome that. And VPU may protect CD4 and so on. And so I think this is something to be thought of and to be experimented. There is at least some reason why you could adapt the proteasomal inhibitors that are used for cancer. Velcade, for example, at low dose, and test it proof of principle. Or use a humanized mouse if you wish to get your initial parameters. There might be small molecules that could antagonize VIF, and I think those should be high priorities. And then there's one technology that I want to emphasize because I do not see it discussed very often. So in the world of small molecules of RNA, so-called siRNA, there's been an enormous explosion of knowledge at a basic level, at a research level to define the function of genes, and there are companies that have been founded to target these molecules for therapeutic use. What they usually talk about in this concept is small interfering RNAs that are dedicated to post-transcriptional gene silencing. And that, I think, is all intuitive to you. You cleave the counterpart messenger RNA, and you use these molecules. But there's another element using small RNAs called transcriptional gene silencing. And I'm giving you a website, which I encourage you to visit. In that latter, promoter-targeted small RNAs can be adapted, and they would have certain features. They would not exhibit transience of the gene knockdown. They would not be affected by saturation of the RNA-induced silencing complex or viral escape mutants. Let me draw a distinction between small RNAs that work at a post-transcriptional gene level and those that work at a transcriptional gene level. The post-transcriptional gene silencing mechanisms work because the siRNA cleaves complementary targeted messenger RNA in the cytoplasm. This is classical RNA interference. I think this should be pursued. But there's another approach that I think has very broad implications, not only for HIV-1, but for cancer and many other diseases. And that is, there's a transcriptional gene silencing. This is well known in plants, but until recently less so in animals and humans. And what you do is you target the small RNA to the promoter in the nucleus, not the cytoplasm. And you may bring about long-term site-specific CpG methylation and, more significantly in my view, a heterochromatic environment as opposed to a euchromatic environment at the HIV promoter, which essentially silences the virus. You don't replicate, you can't start activating the gene unless you have euchromatin. Heterochromatization is a form of sequestering the virus, or any gene that may be there. And so you could develop a system for a durable heterochromatic environment, and that would induce either a medically induced viral latency. The virus is there in the genome, but it's not doing anything. This virus presumably cannot hurt you unless it's replicating. and or you may be able to do a null mutation, if you will, for essential cofactors like CCR5 and so on. So the mechanism of RNA-directed transcriptional gene silencing in human cells is basically mediated effectively in a complex that leads to euchromatin. Here's a few options. It may also be an element of DNA methylation. And this, if you go to the website, which I think is a very interesting mechanism by Morris and his coworkers at Scripps, they have programs that effectively are following what are the semi-durable or durable changes that you could cause at any promoter. And to me, this would be a very dramatic paradigm shift if it could be explored, to essentially trap the virus in the heterochromatin of its host. That's it. It's there. It's silence. We carry around viruses that are in our bodies. Unless they're replicating, they don't hurt you, right? You carry the virus for herpes zoster, many people in their ganglia, right? Unless it's activated, it's not hurting them usually. So, pardon?
I have it now.
Well, that was a high. Well, we need Zostavax, but okay, but it didn't hurt you for much of your life. Okay, so, but again, it supports the theory that it's replicating. That's when it hurts you. If you somehow could silence it, it wouldn't hurt you, right? So at any rate, I think the technology is actually there, but we need the will, we need the incentive, we need the motivation to develop paradigm-shifting approaches. I think the irony is overwhelming to me that I'm now advocating that the drugs that we have are fine, but don't count on them. You must continue the search. And this, you know, the temptation is to say, boy, all these drugs work terrific. What more do we need to do? And I think that would be the wrong choice. I think we've come an enormous way, and I think everybody should be really happy about that. But we should not take this for granted. There are new strains of virus coming in. We have not cut the new infections that are coming into the system. There were just under 3 million worldwide infections of HIV-1 last year, probably in that order of magnitude. And each new infection is an opportunity for a new kind of virus, if you will. So we can't just rest on our laurels. And I think this is an issue. So this gets back to another reason why we can't rest on our laurels. This is the actual report of the new human immunodeficiency virus derived from a gorilla in a human being. Don't take this literally. The patient did not necessarily, and indeed no doubt, one can say without any doubt that she did not acquire directly from a gorilla. That is not what this is, okay? But the gorilla virus, no doubt, has entered the human ecosystem. And someone with whom she had a sexual relationship with was carrying this virus. That person may have gotten it from any of the techniques we were talking about, which involve the hunting and using of chimps or other great apes for a food source, so-called bushmeat. In the process of butchering, there is transmission, there's documented transmission, and so on. So I think this is a real issue, and the transspeciation is still occurring. This was the data actually from the patient. This shows you the viral loads and CD4 count. The only thing you need to pay attention on this slide is several of the assays found it. Was undetectable, including a very common test. So certain commercial tests would have missed this. Other commercial tests actually did get it. So the point is, it's something that requires vigilance. I want to end by going back to the diversity issue. So, and this is something that I think needs to be kept in mind and is one more reason why we need to be cautious here. So this shows you a different way of looking at the antigenic diversity pattern. And this is influenza again as the comparator. There's antigenic diversity. This is a pattern showing little diversity within a host or even within a population at a given point in time, but a relatively rapid change over time, and that's influenza A. In any one time, everybody seems to have the same virus, then it mutates. And that's why you can't use that's why vaccines tend to work in a season, but that's why they're only good for a season, if you see what I'm saying. So at any one time, most people have the same virus, and within an intra-individual variation, there isn't a great deal. But if you look at the pattern of HIV-1, there is substantial within-host diversity over time. And then if you look at various aspects of selection, the there can be enormous diversity with highly diverse cases at both inter-individually and a global level. And this, again, is the same slide I showed you earlier, but it maybe with a different cast. If you look at the right-hand side, you're looking at the Democratic Republic of Congo, you're looking at West Central Africa. This degree of diversity, in my opinion, is very serious, is a very serious element. Within this diversity, there could be new pathogenic strains emerging. There could be strains that are not easily detectable by the current viral load systems. And are we tempting fate that there will be a set of viruses that emerge that don't respond to the drugs? So I've actually lived the irony. There used to be a belief that no drug could work, and that was an obstacle, at least against my lab. Now I detect a subtext that in the long run, no drug can, or at least no drug generally used, the term generally, will fail. That drugs will always rescue us. There will always be another drug available, and it will take on whatever new issue comes in. And I hope that that's true. And there's been remarkable, dramatic progress, no question. But I think it would be unwise to assume that progress would continue. Here's some examples of the continuing prevalence of HIV non-B subtypes. Remember, I said that in Europe and the US, B was the predominant subtype, but not the rest of the world. In Africa and other countries, it may not be. In certain parts of the former Soviet Union, it may not be. So I think that the B subtypes may not be the predominant type in the future. This is my last slide to simply indicate that there is a circulatory effect. New viruses can be introduced. The globe is one globe, and it is possible for anyone from even relatively remote areas of infected endemic areas to be anywhere else in a relatively short period of time. So I do think these are significant issues. I think we need to be optimistic, but we need to be vigilant. Thank you very much.
Questions?
No questions from my lovely wife. She knows too much about this, so she can't ask anything. Please.
So it's kind of a historic question. You started with that in your talk, and you alluded to it a little bit. I'm curious to know, when you started giving AZT, did you expect the ordered accumulation of resistance that was shown not a lot of time after that, and then the steps towards dual therapy and the need of heart? Or when you started, you thought that it was like, you know, a regular antibiotic that would be one drug for
You're asking an interesting question. We did expect that there would be the emergence of resistance, and more often, and actually we acted on that expectation because we never stopped at AZT. And even while the drug was still working its way through the approval process, we had a crash program to develop other drugs that we could bring into the clinic quickly for several reasons. Partially because we believed that no one drug was going to be the solution, and partially because there was a significant fear, in my view, that people would conclude that AZT was a one-drug wonder, was an anomaly, was the exception that proves the rule of untreatability. That, okay, there was a fluke, AZT was around, but it doesn't prove anything. And last but not least, we were disturbed at the signs of over-endowing AZT with more effectiveness and prominence than any one drug could deliver. And so we were very worried about that because we were afraid that that would lead to a reactivation of a kind of what was called by one patient advocate a false hopelessness. And that people had a false hopelessness. You know, you have false hope. Well, there was false hopelessness that nothing could be done. And we were very afraid that that would emerge. I also want to just emphasize that there were significant issues at the time where it's hard to recreate it now. I mean, most people now easily accept that HIV-1 is the cause of AIDS. I think there was an experiment, a political experiment in a major country in Africa where the government, as a matter of policy, decided that HIV-1 wasn't the cause of AIDS and antiretroviral therapy would not be made available. And I think it's fair to say that it had some pretty serious consequences. and that a newer regime has effectively admitted that and is trying to repair the damage done. But it's hard to recreate how prevalent certain aspects of that story were at the time. And if AZT were the only drug around, given its limitations, because some patients can have a response that's relatively durable, but most will have a short-term response. And our fear, quite frankly, was that that was going to reactivate cynicism and fear on multiple levels. And so we felt that it had to be combinatorial drugs. And the only way to make sure that that happened was to essentially develop other drugs on our own. But then the pharmaceutical companies saw that the success of AZT1 was not just going to be an anecdote. And we were able then to work with Bristol Myers Squibb on didanosine or Videx, and we were able then also to work with Roche on HIVID or zalcitabine. We developed those drugs in our lab too, but we were able to work partnerships with them. And the reason they were willing to do that is because they saw there was a success of AZT. And just around all that time, the strong feeling that there were going to be surrogate markers started to kick in. And then the PCR-based things really came in, and that changed everything. Then it became a rational decision for a pharmaceutical company to enter the field. Because they wouldn't have to risk everything, and they could learn early whether things were going to be likely to get approval or not. Very critical for a pharmaceutical company. They need to have metrics for early, either yes or no. But for them, the worst decision is, okay, we don't know, but you're going to have to study this for 5 years or 10 years, and then you find out no. So that's a very tough road for them. But because they didn't have to do that with the surrogate markers, I think that changed everything.
I was wondering about this approach of targeting the proteasomes. I don't know much about that, but what you would assume that would have detrimental effects to normal cellular physiology.
Probably would. So does AZT at certain doses. I didn't stress, but one reason that I personally think AZT was developed was when we were in the Cancer Institute. And I know that sounds difficult, but one, it allowed us to take comparatively unorthodox views for the infectious disease community of the time. It sensitized us to the view that for a serious life-threatening disorder, toxicity is certainly an important consideration, but if you make that the overwhelming consideration, you will ignore the reality that the disease itself is right, mighty, toxic, and lethal. And so we had, because we were in the Cancer Institute, and that was the philosophy that you might have to accept a certain amount of toxicity, we prepared ourselves that might be the case for fulminant AIDS, and we were preparing that. Also, in the cancer world, to some extent, you can argue it's true in the infectious disease world, but less so. In the cancer world, it is truly the norm, rather than the exception, to expect to use combination drugs, even if they must be developed in clinical trials or certainly early feasibility studies as single agents. They're not expected to act as single agents, with rare exceptions, when they're finally used. They're used in combinatorial form for many different reasons, both to avoid drug resistance, to attack the target with more than one avenue of strategic intervention, and to avoid overloading a particular organ, end organ, with any damage from one single agent. So we were preparing to do that. We also prepared for the notion that you're going to get drug resistance pretty fast no matter what you do, and you'll have to prepare for alternatives. I think philosophically that all served us in good stead. The infectious disease community at the time, I have to be careful how I say this, because I don't mean to issue any disrespect of it. It was an understandable position. But their view was still partially informed by the notion that you should strike at one agent. You should find one agent that, if you carefully select it, will work. You should find the agent. And that combination agents, yes, they're used, and yes, good infectious disease people use them, but somehow we were still in that era where you use the right agent. I don't know if you agree with that. And certainly, infectious disease doctors that took care of cancer patients certainly expected to use multiple agents. They expected to use empirical antibiotics when patients had fevers. I understand all that, but still we're at the tail end of an era where, yes, combinations were used for tuberculosis. There were combinations, of course. I know that. Everybody knows that. But there was still a belief somehow that really if you just tried hard enough, there would be one agent. And just try. And that notion that you give up, you're not going to find one agent, especially for an error-prone virus. You're going to have to use 3 agents, maybe 4 agents, to make sure that you don't get emergence, to hit it at multiple points. That notion, at least to me, seemed very, very clear, and I got the impression it was somehow not quite as a high priority in the infectious disease community. I doubt if anyone will agree that that is how they felt. Just as I can find no one who will come forward and say that they believed that the virus was inherently untreatable. I find no one who will admit that they said that, even though I heard them say it with my own ears. But you know, they don't stress that point. In fact, many times they'll tell me they knew it would work all along. These were the same people that would give a meeting talk that preceded my talk that would talk about the impossibility of treatment. So it's an interesting.
When you use 4 agents, does that affect the diversity?
When you use, typically you'd use 3 agents, but if you use multiple agents, you effectively block the virus, you limit its opportunities for escape mutation, and you get durable suppression. And that is why people survive now and why it's a chronic manageable disease. There's no question in my view that that is what's going on. And there's no question that the final recognition, approval, and implementation of what is now called highly effective antiretroviral therapy, which implies multiple agents, that dropped the death rate very dramatically.
When you say suppression, do you still get measurable virus particles?
No, you can actually suppress the virus using highly active antiretroviral therapy, modern therapy. You can suppress it below the detectability of common PCR. You can get back a titer that says we can't detect it. That isn't to say that if you used some highly experimental, nested, complicated PCR technology, you couldn't bring it out. But as a practical matter, the virus in the circulation, RNA virus, goes below the level that you can detect by PCR amplification.
As you approach that limit, does diversity decrease? Like if you normalize the data per number of viral particles?
Well, it does in the following way. You don't get diversity unless the virus is replicating at a significant rate. The virus can't generate diversity unless it's replicating. And it's similar to cancers that way, quite frankly. When you suppress cancer replication, you dramatically reduce the ability for resistant clones to emerge. And you get a very similar effect with HIV-1. If you allow the virus to replicate, or worse, in a way, if you give suboptimal dosing, if for some reason you're giving a insincerely effective dose, if you're giving small doses, or particularly if you're trying with one agent, give small doses or small doses of agent, you will actually permit the virus to select for mutants that are resistant. You give it a chance because its threshold for making the jumps become much less if you do it that way. But if you hit it with very significant multi-targeted drugs, the likelihood of resistance gets very hard. This is why intravenous injection drug users pose an issue. Because for a variety of socioeconomic reasons, they may start treatment, maybe. But then for a variety of reasons, they may not take their full doses, or they may start and stop or whatever, and all of a sudden you've got a resistant strain in them. But they're perfectly capable of transmitting it, either by injection, sharing of needles, or by sexual transmission or whatever. It's not the only mechanism for resistance, but it is a mechanism. And within a patient, don't get me wrong, there are some patients commonly actually who over time will have viral clones escape and will have resistance mutants. That's why you have to genotype them. Not everybody goes down to a level below detectability either. It's that low-level replication. They can be benefited, but while the virus is replicating, each replication is a potential for an error. And the error could lead to a mutation that leads to drug resistance.
Doesn't the survival of those mutants mean that there's a lack of the imposition of constraints? In other words, the host doesn't push them back.
Precisely. This is why the virus is so deadly. The virus has, for its own nefarious purposes, figured out that what it wants to do is first and foremost attack and kill T cells. Perhaps especially T cells that have a high affinity that would bind to the virus in some manner or means. It attacks the central command post of T cells. It likes to kill CD4 cells. Yes, there are viruses that will attack monocytes and macrophages and so on. But the hallmark is a T cell depletion, T4 depletion. So you're actually, if you look at it this way, the virus has evolved to commandeer the cells that could regulate it, push back. But moreover, there are HLA differences. Some individuals have HLA types that can't handle the virus even as well as others. There is an HLA restriction component to this. But it does exactly imply that. Also, the diversity, I didn't want to make it too complicated, but the diversity that you see within a patient, the intra-individual diversity, there is intense diversity, but not all the diversity means that the virus, each virus that's within the person is replication and infection competent, transmission competent, if you see what I'm saying. Some of the viruses may be dividing in the patient and even causing harm in the patient, but they're not necessarily the best ones for a jump from one patient to another. So there is that complexity. There is a replication or reproductive fitness element in all of this. And sometimes certain mutations are not encouraged in the virus because even though it would cause drug resistance, it would also affect the virus's replication fitness. So it's not a simple topic. Please.
Do we have any sort of guesses as to what fraction of the population with these resistance mutations arise due to non-compliance, and what fraction arise just because they arise in a treated individual?
It's a good question. I'm not sure the answer to that. Certainly non-compliance can be a big factor in the emergence of drug resistance. It's not a good thing to do that. If you're going to commit to antiretroviral therapy, it's quite important to make sure that the patients take their drugs. And that's why there has been a very encouraging development of using fixed dose combinations and forgetting about a lot of the different issues that sometimes were part of traditional medicine. There used to be an era where fixed dose combinations were not felt to be that popular. Health ministries didn't like to approve them because they, many years ago, used to have the argument, well, why give a fixed dose? Just give the three things. You know, a fixed dose locks you into a certain ratio and everything. But what people finally recognize, particularly in the developing world, in resource-poor countries, and some parts of the U.S. are like a resource-poor country, you do a fixed dose combination that can be given once a day, and that's your best bet. You do not want the patient to have to talk to the patient. Did you take 6 of your pills? Did you take 4 times a day? Because that's too hard, even with non-AIDS-related medicines. So the people are moving to that. And there's also a tendency in resource-poor countries to use generic drugs, even those that would be protected under proprietary rules in the U.S. The FDA has a process, by the way, of approving. There's a formal process in which the FDA can approve a drug or a drug combination, a generic drug or a generic drug combination, for use in a resource-poor country, even though that combination would not be permitted because of patents or other things or other FDA regulations in the U.S. And that's been a spectacular success because it gives a mark of approval of a health ministry on a drug that can be used in Africa. And one of the more common drug combinations can now be given for $92 a year. I wish it were lower, but still, that's still getting down there. You're not talking $10,000 a year. So that's probably a very strong advance.
What lessons from the developments in these types of treatments do you think vaccinologists can take?
At this point or at the beginning?
as in sort of everything that we've learned in the last 20, 30 years from the development of these treatments can vaccinologists use at this point?
Okay, I think starting at the beginning, the larger lesson is that, particularly if you're going to be making policy, there should be perhaps some element of humility in predicting that an entire avenue of intervention will fail. I think that's one lesson, not only for retrovirologists and vaccinologists, but for anybody. Unless somebody is advocating that they're going to defy the laws of thermodynamics or they're going to violate some established principle of physics or chemistry, people should be allowed, if possible, to outline the premises by which they expect to succeed with milestones that would be necessary to confirm or reject the hypothesis that they're putting forward. And with an opportunity, as I mentioned in some of my comments, where possible to have the same investigators serve as both a laboratory and clinical operation. That was true in my laboratory. As I mentioned to some of you, we had a lab, and then a few feet down the hall, we had a clinic that we could traverse and that we were both. And we, therefore, didn't have to argue with quite as many committees. I would say we didn't have to, we were not exempt from arguing with any committee. But besides a few ethics committees and things like that, we were responsible for our own potential failure. We could execute a plan and deliver it and didn't have to importune some other agency to please implement this. Or we didn't have to deal with pharmaceutical companies that were uninterested in this problem and felt that there was no hope. We were able to form relationships with those that felt there was an opportunity. So I think that that's very important, if possible, to have people around who can wear both hats. And I see that actually is going the other way. I see there's a division of labor, and there's laboratory people and clinical people, and they're not necessarily the same. So I think there's an element of that. In terms of vaccinologists, now my argument for them today would be don't despair, don't give up, don't declare things are impossible. Try to have an even keel. By the same token, stick to the data. Don't torture data until it confesses. Basically, it's very important to be objective, even if you're really committed in the deepest way to a particular project, and that's very hard to do. But I think that's critically important. So I think don't despair, don't declare vaccines impossible, but recognize that there's a long road ahead for them.
Between HIV and influenza. In influenza, there's a hemagglutinin, so it's possible to have a protective vaccine.
At a given season.
Why?
Because your vaccine for the year 1998 probably will not work too well for the year 2009, even for seasonal flu, classical seasonal. That's why we need to do a vaccine every year, right? The vaccines change, but everybody gets the same strain at any time.
Why has a protective vaccine against HIV been so elusive?
To me, it relates to two things, one of which you said. One is the tremendous diversity. So if you speak in teleological terms, and you were willing to allow the literary device of letting the virus think about what it wants to accomplish, and then have the media be the evolution would do the rest. If you're a virus and your goal is to jump into humans and replicate yourself, there are two things you have going for you, very dramatically. One is you have an error-prone virus, replicating enzyme, the reverse transcriptase. So that constantly generates mutations for you, gives you a wealth of mutations, any one of which can then take off, any one of which can be an immunologic escape mutation. It frustrates the immune system merely on that basis alone. But the second thing you do is you take out the cells that are probably responsible or bear a high role in containing you. there are lots of examples of retroviral infections in human beings that were foiled, if you will, and that became dead end. They became molecular fossils. The genome was composed of what? 8 to 10% of retroviral elements? You guys are genome scientists. I'm pretty sure that figure is about right. Okay, so 8% or 10% of the genome is made up of fossilized viruses, retroviruses. Presumably they're dead ends, right? We're here. Now you could argue we got bottlenecked and whatever, but I like to think we're still here. So there is evidence evolutionarily of a constant war of evolution between pathogenic retroviruses in primates and host-respective enzymes. The ApoBec series got very significantly amplified, the family of genes that we call the ApoBec, the editing genes that I described for you. There's one copy in mice, and there's many, many copies in different family members and paralogs and so on in humans. And so you can see evolution has kind of expanded those. So there's been a war back and forth. And the virus, the most effective virus that we currently face is one that gets in, is highly diverse, becomes diverse quickly, and kills the central T cells that are sent to defend against it. Possibly with the T cells that have the greatest likely affinity for it going first. Maybe. If you see what I'm saying. And the virus is cytopathic for T cells. It destroys them, there's no question. Clinically, you can deplete your T cell count down to...
If it were diverted, I mean, if there were a way to divert it, let's say, to phagocyte.
Pardon?
To another population of cells.
There are viruses that have tropism for macrophages, definitely. They don't kill the macrophage. Unfortunately, they're a reservoir.
So you think they would end up in T cells anyway?
Yes, I don't think that. I know that. I mean, that's shown in different experimental models, but there are microglial elements, for example, even in the brain, that give you a reservoir in the brain, and the virus just periodically infects new cells. Every time there's a monocyte or macrophage contact with its coordinate T cell, there's an opportunity for the virus to jump. But it's no question that there are some tropic strains. We've studied them. One of the first questions we had to ask is, will the drugs that work against T cell tropic viruses work against monocyte tropic viruses? Because one of the fears we had was, you could just interfere with the T cell tropic viruses, leave the monocytes alone, and then constantly have a reservoir in the patients. And what we found is very quickly that the antiretroviral drugs worked. They didn't always work in the same priority. In other words, sometimes a dideoxyinosine would be more effective than an AZT, depending on what target. But this general class worked in monocytes just fine. Carlo Perno in my group showed that very early on. So we were very confident that we weren't going to have that artifact. But it may not necessarily be cytopathic in the sense of destroying the monocyte. It just may be that it's a nice reservoir for it.
I don't know if it's too late, or should we go?
Go. Sure.
So just one comment about the discussion previously. So there is work on ongoing replication and generation of mutations and undetectable viruses, and I'm just mentioning it because, interestingly, the person who's doing some work on it is the daughter of Hiroaki Mitsuya. She was she's now a medical student, but she was at Stanford University working with a group where I did my postdoc, and they had some work showing that there is evolution of resistance, although, like you say, it's much lower than when the viral load is high, but there is some evolution of resistance, and she did some work on it. So this is very interesting.
Well, there certainly is a potential for the emergence of resistance in anyone. It's a probability effect. So with the highly active drugs, particularly if you combine multiple stages, the nucleoside analogs like AZT or adenosine or so on would tend to hit at the conversion between reverse transcription from RNA to DNA or from the first strand of DNA to the second strand of DNA. Probably that's a major point before the process hits the nucleus. The protease inhibitors would hit it at the other end. So the protease inhibitors actually would hit it at the point in which an infected cell is packaging virus for export. So when you combine them, you're hitting it at both ends, and the protease inhibitors can, because of the combinatorial effects, among the things they do, they can dramatically drop the titer very quickly when you use them together. They can give you a very dramatic drop because you're blocking that end stage. There isn't a pipeline effect. You see what I'm saying? You're the final packaging. It's like if I had something that blocked the stuffing of envelopes for FedEx packages, but the package is already sealed and ready to go, it would still go out. It would take me a little bit of time before I saw the drop in FedEx deliveries, right? But if I interrupted the packages before the FedEx guy could even pick them up, but just at that stage, then you would immediately see a drop in FedEx deliveries, right? You like my little analogies? Soren loves them. So, and because you then are inhibiting it at multiple ends, you get very dramatic drops. You're seeing drops among HIV that people, frankly, do. Now they believe it, but there was an era where there was no one predicting it. There were so many prophecies that have been confounded in this field. It almost is worth writing them down someday, because basically there was all the 5 prophecies I gave you. Okay, then there was a whole bunch I didn't give you, which I'll give you now, including that the technology, okay, maybe you can get away with this in a high-tech Western country, but you can never have an effect on mortality in sub-Saharan Africa. Wrong. Absolutely wrong. Even in poor countries like Malawi, you can see by the scale-up of the drugs have to be cheap and they have to be available. Of course, you're not going to see effect if you're charging $15,000 a year. But if you have them available, the death rate drops very quickly, sometimes in the year of introduction, in Malawi. So now there are differences. You probably don't get the same kinetics of a drop in mortality as you do in Western countries because you have to worry about TB and you have to worry about other. But there is no question that you're getting an effect. So the notion that the high-tech thing couldn't be applied in Africa is wrong, right?
And they won't be adherent?
They won't be adherent. Absolutely the opposite. There are people in Africa that are more adherent than some people in Baltimore. It is astonishing, and primarily because many of the people in South Africa or various Namibia and what have you, they have a virtue of wanting to live. And they believe the doctors, and they will. I mean, Bob Gallo came back and told me once that he was greeted by people, survivors of AIDS, who were dancing in the streets at his arrival. And I reminded him of 2 things. One, that usually people who are dying don't dance in the streets. And I said, well, Bob, you probably got it mixed up. They were dancing because they thought you were leaving. But that's a little private joke. And so he, in that sense, can take a lot of satisfaction because he proved the causation of HIV-1. That all of this is predicated on the causation, knowing the causation. If there was doubt about the causation, then it would have been impossible for us to begin any clinical programs. We could not be injecting chain-terminating nucleosides into people who were very, very sick because a retrovirus was one of 100 candidate infectious agents that might be the agent. Or there might be many, many different causes. You could not conduct a program based on that. So causation was very critical, not only to prevention and blood bank screening and whatever, but it was very critical in the arena of developing novel drugs. And you can't, in my opinion, you can't develop drugs in this context without knowing causation. That's my view.
So the question I had to do with the non-B aspect that was at the end of your talk. So I'll ask the question, but I'll first state that, we're actively working on this now. We're collaborating on this aspect directly of non-B, specifically on HIV drug resistance. So
well, I hope you found the talk useful in that context.
Very, very useful. So it's very relevant, and we have a lot of international collaborations and are aggregating data to address exactly that. So I guess my question is, taking all that in mind, what is Celera's sort of view to the future, and how do you apply sort of that aspect of continuously evolving virus? How do you apply that to the future?
It's a very interesting point. Celera, I give you a disclosure, has a working relationship and alliance with Abbott. And we are particularly commercially developing what's called the M2000 platform, which is an RT-PCR system, as you know. It has a deliberately tolerant probe design, so it accepts mismatches, particularly at the integrase. And I will make the following statement as austerely as I can, so that I'm not in any way attempting to promote any product. The Roche product, factually, by authors who do not work for Celera or Abbott, failed. I showed you the data. I did not do those experiments.
Oh, I know
that was how it was reported.
It's almost a known...
The M2000 platform picked it up. Draw your own conclusions. And so if you say, what is Celera doing? It is attempting to make sure that it keeps up with the technology and that these non-B subtypes, and particularly the problem of transspeciation, which I think could be, I don't know. It's the future. It's the unknown of the future. I don't know whether transspeciation, it's an embryo. I don't know whether it's going to be a mouse or an elephant. They all look the same at a certain stage of development, right? And so I don't know that transspeciation is going to be a huge problem, but I am concerned about it. I think the conditions for transspeciation exist in Africa. I think many areas that were previously totally inaccessible now have roads that go to them for logging. I think you have logging camps in areas that were essentially forbidden except to certain local, regional individuals who know how to survive in them, and now anybody from any urban area can make it. I think you have loggers. You have sex workers who are following the truckers and the loggers into the camps. And you have bushmeat. Maybe that's not a perfect storm, but it's a semi-perfect storm. So you're bringing all the factors in that normally would have been segregated and not developed, and you're bringing all the factors to have. So the chimp or other kinds of primate viruses, are they human retroviruses in waiting? You know, so we don't know. And I think we'll need to have screening technologies that can deal with it. And I think clinical labs that are using technology that might miss non-B, clinical labs, need to understand that this issue is more than theoretical. What they choose to do about it is their call. But my point is that this is not a problem of such rarity or anecdotal nature that it's okay to say, well, if I'm missing. There are clearly there was a report, as you probably know, very recently of a group G that was published electronically in advance of actual publication that was missed cold. And the doctors kept checking back, and they missed it, and the patient actually deteriorated clinically in front of their eyes. There's a group in Florida at the University of Miami that found a group F that was missed. So I think these are issues that are real, and as these non-B subtypes come in and as other transspeciations occur, I think this could be an issue. That's why I'm a little bit worried about it, and that's why, to be candid, I find it ironic that I'm worried about it, and a lot of people in AIDS don't seem to be, because they think the problem is, okay. They don't exactly act blasé, but they don't have that sense of fire in their eyes that we had in 1985.
We had a case here of HIV-2 that was missed, and we had to use different assays in order to show that it was that.
So I think that's very important. I think typically what happens, not typically, but I think it's not rare for the following to happen. The physicians think the patient is deteriorating with HIV or some related syndrome. They send the thing to the lab. The lab says no titer. They write back saying, listen to us very carefully. Read our lips. This patient probably has AIDS and is deteriorating. Do the test again. And they do the test again, and they say no titer. And they say, the physicians say, listen, you didn't hear us the first time. The patient probably has AIDS. So if you're telling us you don't have AIDS, it's your problem. And then somebody, probably you or somebody, finally says, okay, well, let's get serious about this.
We have to use a different answer. We actually published it as a case report.
Maybe I'll give you my card. You can send it to me, because I've been tracking these. But yeah. So I'll give you my card. You can email it to me.
Sure.
But I think it's going to be more than an anecdotal problem. I think this is probably going to be the future. I think that we're going to find The viral load assays are going to be not necessarily up to the task. If we get new transspeciation, it could be an issue. And we're taking a lot of stuff for granted. I hope that it's simply an overreaction. I'm hoping that it's just that this is not going to be a big deal. But just because the death rate dropped dramatically, and notice it did not go down to 0. It dropped. It's a very gratifying drop, but there was still a plateau. And so there's that issue, constant issue. But I think the other issue is if you get new transspeciation, or the other thing I didn't get into is chimeric strains. HIV-1, as you know very well, can recombine in people. It isn't just standard genetic diversity, it's actual recombination. And it could recombine and become even more virulent. I think we're asking for a lot. If we don't keep on top of this and keep ahead of it, I don't know, does the NIH still give it the same priority?
We have the collaboration that I told you is a product of an R01 that I have to study HIV drug resistance to multiple subjects.
Yeah, but that might speak more about you and your capacity as a scientist than the priority.
I give a lot nowadays for anything, so it's hard to answer.
It's hard for me to understand that. You're not the only person who's told me that, but there was a $5 billion stimulus package. That's not.
That was for U.S.-based. I submitted things to that. I didn't get anything because it was based on international aggregation of those recombinants and those diverse subtypes, and they wanted U.S.-based money to go to workers in the U.S. That was their focus. So
I have to say that is profoundly. That is profoundly profound. I mean, if somebody's telling me that HIV in Africa would not be an American problem because, after all, it's Africa and not America, that I would have to say is a very complex line of thought. Besides, I'm not averse to job creation, and that's all wonderful, but that can't possibly be the primary impetus for NIH spending. It cannot possibly be, because it has to be that people are given NIH funds to reduce premature deaths or alleviate suffering. That has to be the prime, or at least a logical opportunity to extend to that point. And in theory, you should use the best means of doing that as possible. If you adopt any other posture, think about what you're saying. Then the NIH is a modified version of an entitlement or a post office or something, right?
Right. So does Celera have interest in research and sort of pursuing?
Well, Celera through the Abbott Alliance might. John Hackett, of course, whom you know, no doubt. John is a formidable intellect and thinker, and he's someone who I'm sure would be. I mean, send me an email, I'll give you my card, and it may be possible to put you in touch with some people. But most of our work would be under the umbrella of the cooperation between Celera and Abbott.
I was in touch with Mark Samber, I'm sure you know him. He's the Global Scientific Affairs Director.
But John Hackett is an actual very high-grade scientist in his own right and is specifically interested in these issues. And you might also want to form a collaboration. I don't know if it's effective, but I wouldn't think it's impossible to form a collaboration with Gallo's group at the Institute of Human Virology, with Bill Blattner, one of the people there. At a minimum, he might be able to get you interesting samples and so on. And Bill goes to Africa, as no doubt probably you do as well, but he goes there quite often.
Rami is a close collaborator, so it might fit into your help to connect.
Yeah, I think this is a very important area. It should be a high priority area. I really would be surprised that they wouldn't fund you because you're. I mean, it's an American problem. It was the #1 cause of death in young people not that long ago. It could happen. It could come back.